In 2012, two scientists discovered a tool that lets us rewrite the code of life itself. It's called CRISPR, and it didn't come from a lab dreamed up to edit genomes. It came from bacteria, defending themselves against viruses for billions of years. This is the story of how an ancient immune system became the most powerful gene-editing technology humanity has ever built. Long before humans discovered it, bacteria were using CRISPR to survive. Viruses called bacteriophages constantly attack bacterial cells, injecting their DNA to hijack the host. But some bacteria fight back. When a phage attacks and the bacterium survives, it cuts out a snippet of the invader's DNA and files it away in its own genome, in a region called a CRISPR array, a genetic mugshot book. The next time that same virus attacks, the bacterium recognizes the sequence instantly and destroys it before infection can take hold. For decades, CRISPR was just a scientific curiosity. Then in 2012, biochemists Jennifer Doudna and Emmanuelle Charpentier made a breakthrough discovery. Studying how bacteria use a protein called Cas9 to slice viral DNA, they realized something extraordinary: this natural defense system could be reprogrammed. By pairing Cas9 with a custom-designed guide RNA, they could direct it to cut any DNA sequence they chose, not just viral DNA, but any gene, in any organism. In 2020, Doudna and Charpentier won the Nobel Prize in Chemistry for turning a bacterial immune system into a universal gene-editing tool. The CRISPR-Cas9 system has just two working parts. The first is Cas9, a protein that acts like a pair of molecular scissors, capable of cutting through the double strand of DNA. The second is the guide RNA, a short synthetic sequence that scientists designed to match any target gene. The guide RNA locks onto Cas9, and together they go searching through billions of DNA letters, 3 billion base pairs in the human genome alone, for one exact matching sequence. Finding the right spot isn't guesswork. Cas9 scans DNA for a short marker sequence called a 𝗵𝑚, found next to millions of locations in the genome. Once it finds a 𝗵𝑚, Cas9 unzips the DNA double helix beside it and tests whether the guide RNA matches the exposed strand, letter by letter. If it matches, the RNA locks in, forming a structure called an R loop. Only then does Cas9 commit, clamping down and preparing to cut precisely at that one location, out of billions of possibilities. This is the moment of the edit. Cas9's two cutting domains, HNH and RUVC, slice through both strands of the DNA helix at once, creating what's called a double-strand break. To the cell, this looks like catastrophic damage, a genome-threatening emergency. And the cell's emergency response to that break is exactly what makes CRISPR editing possible. The cell has two ways to repair a double-strand break. The fast, sloppy option is called non-homologous end joining. The two broken ends simply get glued back together, often deleting or inserting a few random letters in the process. That's enough to disable, or knock out, a gene entirely. The slower, precise option is called homology-directed repair. If scientists apply a DNA template alongside Cas9, the cell copies that template to heal the break, allowing researchers to insert, correct, or rewrite a gene exactly as designed. This precision is already changing medicine. In 2023, the first CRISPR-based therapy was approved to treat sickle cell disease and beta thalassemia, editing patients' own bone marrow cells to switch a healthy gene back on. Researchers are engineering CAR-T cells to hunt cancer more effectively, developing CRISPR treatments for inherited blindness, and testing in-body edits that could one day correct genetic diseases with a single injection. CRISPR's reach extends far beyond human medicine. Scientists have edited crops to resist drought, disease, and browning, without inserting foreign DNA, creating gene-edited produce that regulators treat differently from older GMOs. CRISPR-based diagnostic tools like Sherlock can detect viruses, including COVID-19, in minutes using the same molecular targeting system. Researchers are even exploring CRISPR to fight antibiotic-resistant superbugs and preserve traits in endangered species. With this power comes serious responsibility. CRISPR isn't perfectly precise, it can sometimes cut unintended, similar-looking sequences elsewhere in the genome, called off-target effects. And in 2018, a scientist in China defied international consensus by editing human embryos that were brought to term, creating the first gene-edited babies, an act condemned globally as reckless and premature. Editing genes in eggs, sperm, or embryos changes not just one person, but every generation after them. That's why most of the world's scientists agree, germline editing in humans must remain off-limits until safety and governance catch up with the science. From a bacterial defense system, to a Nobel Prize, to a tool now curing genetic disease, CRISPR's story is barely a decade old, and it's still being written. Cheaper, faster, and more precise gene editing tools are emerging every year. The technology that once just helped bacteria remember their enemies now gives humanity the power to rewrite the code of life itself. The science is here. What we choose to edit, and what we choose to leave alone, is up to us.